Greetings, viewers. Welcome to the online resource portal hosted by Haldia Institute of Technology. Our topic of today's discussion will be about hydrogels, the insights and future prospects, brought to you by the Department of Biotechnology, Haldia Institute of Technology, and delivered by Dr. Chetan, Assistant Professor, Department of Biotechnology. So let's begin the session, shall we? Starting off with the introduction to hydrogels, a hydrogel is basically defined as a type of soft material that is formed by cross-linking hydrophilic polymers and is capable of expanding in aqueous solvents and retaining water. Studies related to hydrogels were reported for the very first time in 1874. By middle of 20th century, hydrogels were classified and termed as polymeric networks that had cross-linking and were swollen because of retaining water within the network as seen in the figure. Since then, as seen in the images to the bottom left, there has been an exponential increase in the publications and interest related to hydrogel research. The image on the left shows research with different areas of regenerative medicine that reported some studies related to hydrogels. Moreover, the one beside it shows the exponential increase every year recorded in the number of search queries associated with hydrogel research. Now, taking a look at some of the common mainstream applications of hydrogels. It can be used as an excellent absorbent material due to its native fluid holding attributes. Contact lenses have been reported to be made up of polymeric hydrogels. Gelma, a very superior grade hydrogel, has been actively deployed in wound healing where drugs or stimulating agents are trapped within the cross-linking network and the hydrogel and feature controlled release, thus stimulating tissue regeneration as shown in the figure. Similarly, cells can be trapped within the cross-linking network and successfully immobilized with minimal damage. Cells can be even retained and transported without losing any viability if the hydrogels deployed are specifically engineered with cellular growth and maintenance media. A very specific application of cell immobilization is found in tissue engineering scaffolds, where stem cells are captured within hydrogels and transported to the target tissue for stimulated growth and regeneration of tissues. The word SMART for hydrogels was introduced in 1948 by Kuhn and co-workers. Delivery concept denominated as SMART because it can detect prevailing stimuli and respond through structural, morphological, or functional changes. Their unique properties can be associated with environmental factors such as pH, enzyme, ionic charge, glucose concentration, heat, light, magnetism, electric charge, and even mechanical stress. The first publication about polyacrylic acid or PAA molecules shown in the diagram below that could undergo structural adjustments in accordance with the media pH were reported in public domain for the very first time. The dynamic nature of supramolecular chemistry makes it applicable to the development of smart hydrogels. The final morphology expected for the hydrogel is associated with a series of variables which primarily include the synthesis or treatment procedures of original polymer, monomer composition, and the ratio as demonstrated with example of a dynamic drug delivery system below. Now coming to classification of hydrogels. Basically, hydrogels are classified on the basis of origin, degradability, and cross-linking mechanism. However, these factors and some intrinsic properties of hydrogels such as fluid involved etc. are often manipulated or engineered to control the structural properties of final system. The table here shows details about how supramolecular interactions and the dynamic covalent bonds are engineered to get hydrogel system with desired properties. When successfully deployed, the changes can result in degradation properties, drug release, swelling changes in shape or surface, conformational modifications, or micellization to the user's liking. Based on these inputs, we can schematically draw a very fine line between hydrogel structure and smart materials affecting the properties of native system and determine the responsiveness of final hydrogel system as shown below. So now let's discuss the thermoresponsive kind of hydrogels. These hydrogels came into being out of sheer necessity of fluctuations in physiological temperatures and differences in the living organisms. Their applicability is what makes them most studied and engineered hydrogels spanning regenerative medicine and drug delivery system. Generally, there are only two kinds of thermoresponsive hydrogels. The lower critical solution temperature, abbreviated as LCST, and upper critical solution temperature, abbreviated as UCST, as shown in the phase diagrams below, plotted as temperature versus polymer volume fraction. Just for sake of some figures, Google Scholar reported only around 44 publications for UCST and over 300 for LCST, which clarifies the workability and applicability of both classes of thermoresponsive hydrogels. As seen in the figure, it is very easy to comprehend how these hydrogels would interact with a change in temperature. Several polymers can be included in both LCST as well as UCST classes. For example, PEO, which shows a loop-shaped miscibility gap with UCST greater than LCST. For LCST hydrogels at lower temperature, water molecules are arranged around the polymer. The increase of temperature reduces the energy associated to the water-polymer interaction. Thus, the polymer dehydrates, transforming itself into a more hydrophobic structure and causing phase separation as shown in the top left corner. Table 2 shows molecular structure of some thermoresponsive polymers and how they would interact with temperature shift. Moreover, phase transition temperature of some common LCST and UCST polymers has also been listed, along with applications of such thermoresponsive polymers in drug delivery systems, tabulated as Table 3. Here we see how hyaluronic acid engineered hydrogel system behaves when temperature is greater than CGT. Apart, a hydrogel based adhesive system is shown that is functional only at body temperature. Hematoxylin and eosin strained in vivo micrographs exhibit the applicability and regeneration through thermoresponsive injectable PNI-PAAM hyaluronic acid copolymer hydrogel system for adipose tissue engineering, and tissue growth is visible at all proper locations. Electric, magnetic fields and light exposure are main external physical stimuli that can be used to tune drug release kinetics from smart hydrogels. Once this stimulus can be controlled, they have an advantage of modulating drug release spatially as well as temporarily. Photosensitive polymers can change their physiochemical properties or degrade in response to light irradiation of appropriate wavelength and intensity. There are basically two types of UV light response. First, photopolymerization, and second, photocleavage, as shown in the figures. Now, there are two main disadvantages of using UV light as reported that are related to its low penetration due to its intrinsic properties associated with wavelength and the fact that it's carcinogenic. Some researchers have found an alternative to UV light where we use UCNPs, which are able to convert photon energy absorbed from NIR light into UV light in localized form as shown in illustrations, thus eliminating all negatives of using UV light. Now coming to charge and magnetism responsive hydrogels, they are classified as hydrogel systems that can change their properties under influence of ionic or electric charge or magnetic field. They are termed as electroconductive hydrogels, ECHs, developed from polyelectrolytes or from ICPs. Here we have structure of some of the conductive polymers such as polyacetylene, polyphenylene vinylene system, polypyrrole and polythiophene system, and polypyrrole and polyphenylene sulfide system. Below is the schematics of how charge and magnetic field affects the properties of hydrogels and its applicability in drug release system. The development of magnetic nanomaterials has been the source of discovery for spectacular new phenomena with potential application in numerous fields including bioimaging. The HFMFs are less invasive than electric fields or light exposure, and magnetic hydrogels developed by inclusion of MIONs in the polymer matrices have found their application in many fields. The MION's polymer ratio is what determines the saturation magnetization and consequently the drug release performance as shown below. Just like thermoresponsive hydrogel systems, pH responsive systems were fabricated due to the fact that human body exhibits substantial pH changes in different body parts when functioning normally as seen in the physiological map below. Additionally, some diseases can also cause pH changes in human body. Similar to macromolecules, a polymer network contains ionizable groups that can donate or accept protons in response to pH. In aqueous solutions, they are polyelectrides, and their relation with pKa values is illustrated. Basically, there are two types of pH responsive hydrogels, namely polyacids and polybases as listed below. Table 4 shows several applications of pH responsive hydrogel systems. Apart from chitosan and alignate description as pH responsive hydrogel systems, we have an illustration here that shows difference between pH dependent water permeation through porous and a dense membrane. Important feature of hydrogels considered that some biological processes such as nerve excitation, muscle contraction and cell locomotion involve ionic strength modification. Generally, the mechanism of ionic strength responsive polymers is that the added salt will reduce the electrostatic interactions between copolymers or between polymers and other molecules as listed or illustrated below. Some weak PAAs and MAAs shows increased degree of ionization upon addition of lithium methoxide salt. Hydrogels composed of PNI-PAM exhibit LCST. Its behavior also showed a sharp volume phase transition depending on the NaCl concentration as well. The PNI-PAM hydrogel with specific sensitivity was synthesized by introducing dibenzo 18 crown 6 monomers in network as shown in the schematic illustration. Changes might occur in response to the increased concentration of specific biomolecule availability. Antibodies, T cells and enzymes are important biomolecules that can be used as important signals for disease diagnostics. Many research groups have focused in the development of smart self-regulated biomaterial systems used for drug delivery as well as diagnosis, cell cultures, biosensors or matrices for tissue engineering and regenerative medicines. There are three types of responsiveness associated with glucose, enzyme and antigens. The glucoresponsive systems are basically used in diabetes treatment with different strategies for insulin self-regulation, such as glucose oxidase based enzyme self-regulation, lecithin covalin A, phenylboronic acid based enzyme regulation. Now coming to GOX based enzyme regulation, we can see that glucose diffusion triggers the reaction, as a result of which an enzymatic reaction converts glucose to gluconic acid, thereby resulting in increase in gel volume and decrease in pH. This can be used to trap insulin within the hydrogel system and release it with glucose as trigger or stimuli. Here we can see lecithin covalin A in action, where addition of glucose occupies interstitial positions within the hydrogel framework and the mutual interaction with native molecules results in increase of the gel volume. Here we can see PBA in action, where a blue shift in color defines shrinkage and the red shift for swelling. These all approaches are basically to target insulin self-regulation using hydrogels as carriers. As of latest reports, novel dynamic covalent bonds have been fabricated that have better efficacy than discussed legacy approaches based on GOX, lecithin and phenylboronic acid or PBA. In cellular environment, most stimuli responsive mechanisms are under control of enzymes. Since enzymes are highly selective in their reactivity, the responsiveness is very specific. First application related to tissue engineering for cartilage repair and chondrocyte culture was with PEG based hydrogel system that exploited its specificity to trigger volume changes. PEG structure is illustrated here. Enzyme responsiveness is also widely exploited to release drugs, specifically in the colon, where enzymatic reaction or the activity is at very high rate. Biodegradable polymers include naturally derived polymers such as pectin, amylose, gelan gum, chitosan, alignate and dextran. Artificial non-biodegradable polymers include PEG, PEO, PHEMA, PNIPA, etc. Antigen responsiveness hydrogels are able to undergo volume or structural changes in presence of antibodies. And finally, the generalized functional delivery systems. The applications of these bioactuating systems requires a sophisticated molecular design which increases their potential applications. Some diseases are known for significant changes on the homeostasis state as well. Pathological aspects that can be explored towards this objective are: first, inflammatory processes, cancer therapy, diabetes mellitus, etc. And now to conclude, the biggest question about the future prospects. Which applications of smart polymers or hydrogel systems will likely succeed in clinical trials or applications, and why? Answer 1. Tissue engineering. Point number 1. Because it's harder to stimulate bodily environments for tests. Next, it's difficult to assess performance of grafts, scaffolds, and other devices in vivo. May be developed for longer term applications, but in turn it will take longer to test the materials when used with hydrogels. It depends on whether you want to implant the polymer scaffolds with cells or wait for scaffolds to degrade before implantation and when cells take over. Second application, drug delivery. It's a short term operation, so it takes less time to complete testing. Simple methods are available to study release in vitro and able to test the drug concentration for in vivo tests as well. And finally, smart diagnostic applications. Hydrogels can be used as testing materials, if not in the body, regulations are much easier to meet and test. This brings us to the conclusions of today's session. Hope you enjoyed the session. Thank you for your patient listening.